My name is Denise. This is a PhD dissertation on the development of human valvular calcification via fluid-induced oscillatory shear stress-based cell and tissue culture. The cardiovascular system consists of the heart and blood vessels, which are responsible for nutrient transport and waste removal. Blood vessels consist of vascular endothelial cells and vascular smooth muscle cells. The vascular endothelial cells are subject to flow, which can be laminar or oscillatory. Flow oscillations are often found at bifurcation sites, and diseases such as calcification or plaque formation can occur due to improper cell communication from hemodynamic factors. Heart valves provide unidirectional blood movement from the ventricles to systemic body. Valves consist of valve endothelial cells, or VECs, on the surfaces and valve interstitial cells, or VICs, on the inner layers. Similar to blood vessels, valves are also subject to flow. Flow oscillations are often found on the fibrosa side of valve leaflets, while the ventricularis is mainly dominated by laminar flow. Diseases such as calcific aortic valve disease can occur due to improper cell communication from hemodynamic factors. The aortic valve market is estimated to have an increase in 13.2% between 2020 and 2030, with Europe being the largest and fastest growing market by region. One of the main reasons for this market growth is an increase in calcific aortic valve disease, or CAVID, which is one of the most prevalent chronic heart problems with over 100% growth rate in the last 30 years. Common problems associated with CAVID are valve stenosis, increased regurgitation, and reduced cardiac output. Current treatment options for early and intermediate stages of CAVID are not available. Severe treatments include bioprosthetic or mechanical valve replacements. Bioprosthetic valves are prone to recalcification and may need multiple replacements, and mechanical valves require patients to be on lifelong anticoagulants. Both cases are invasive and therefore are limited to a selective patient subset. In terms of drug therapy, current available therapeutic targets for vascular calcifications are ineffective in treating CAVID. Therefore, understanding similarities and differences in the responses of valvular and vascular tissues due to flow oscillations can help identify future CAVID treatment options, particularly valvular pathologies that are less studied in comparison to vascular diseases. Previous studies in our lab, we found a sweet spot OSI for valve tissue growth. But first, let's take a look at what OSI means. OSI stands for Oscillatory Shear Index and is a measurement of flow disturbance. Its equation quantifies the ratio between forward shear and total shear, where big T is duration of cycle, tau is the wall shear stress, and small t is time. The OSI value ranges between 0 and 0.5. This is a visual representation of OSI and flow. We can see that when OSI is 0, flow remains in static or steady. At OSI of 0.25, there is moderate oscillation, and at 0.5, flow is at full oscillation. Previous studies in our lab conducted by Dr. Alex Williams, we found a sweet spot OSI for physiological valve tissue growth, and valve development gene expressions were upregulated within the sweet spot values. Other studies have shown that bidirectional flow induces VEC phenotype change through reduction in gene expressions that regulate cell phenotype transitions and inflammation. This phenotype change often leads to endothelial dysfunction. Under physiological flow conditions, we also know that low wall shear stress is generally accompanied by high OSI. This combination also promotes atherogenesis. The objective of this study is to investigate how quantity of fluid oscillations using the OSI parameter induces cardiovascular remodeling. We hypothesized that high OSI modulates cardiovascular phenotypes to induce remodeling. There are two systems that we are interested in examining. First, we looked at the effects of OSI on vascular cell types. Our aim 1 is a non-biased approach that evaluates the extent to which paracrine signaling from vascular endothelial cells exposed to low, moderate, and high OSI conditions can alter vascular smooth muscle cell gene expressions. First, we purchased commercially available human aortic endothelial cells and expanded them in flasks with the appropriate growth medium. The vascular endothelial cells were then conditioned in a bioflux shear assay system using 24 well plates that consist of 8 microfluidic channels per plate. A consistent shear magnitude of 1 dyne per centimeter squared was applied to the endothelial cells for all flow groups: static, steady flow, 0.25 OSI, and 0.5 OSI. And each group was subject to a conditioning time of 48 hours. Next, we purchased commercially available human vascular smooth muscle cells and expanded them in flasks with the appropriate growth media. To simulate paracrine signaling, we used the conditioned media from endothelial cells in all flow groups to further culture smooth muscle cells in static for 7 days. 50% of the media for smooth muscle cells were the previously collected conditioned media from endothelial cells in the bioflux plates, and the other 50% was fresh media. We collected the RNAs from both vascular endothelial cells and vascular smooth muscle cells and delivered them to University of Miami for RNA sequencing. Raw RNA sequencing data consisted of FASTQ files, which we then collaborated with Dr. Yu's team from the Department of Biostatistics for alignment of base pairs, mapping with gene cards, and totaling the number of raw counts for each gene found. Over 50,000 genes were identified in the sequencing. With help from Assad, we then computed the fold change, constructed a graphic presentation, and identified significant genes expressed across the flow groups for each cell type. We conducted 3 samples per flow group for each cell type. The graphs shown here consist of dots where each dot represents a gene. The redder the dots, the lower the p-values, and the more significantly different there is found between the groups. In the case of endothelial cells, most significant differences were found between the static and the 0.5 OSI groups. The most significantly different groups for smooth muscle cells were the steady flow and the 0.05 OSI. The bluer the dots, the greater the p-values, and the more similar they are between the groups. In the endothelial cells, the most similar flow groups were steady flow versus 0.25 OSI, and in the smooth muscle cells, the most similar flow groups were 0.25 OSI and 0.5 OSI. For the endothelial cells, here are the top 15 significant genes between flow groups in order of highest significance. Future studies involve further mining of this data to understand relation between flow, OSI, and gene expression. There are some interesting genes here, as pointed out by Dr. Hutchison earlier this week during our meeting. specifically piezo-2, which is a mechanosensitive ion channel that won the Nobel Prize Medicine in 2021. Thank you, Dr. Hutchison, for sharing that information. I myself have been a little out of date on Nobel Prizes these days. The last award I read on the newspaper was back in 1921, when Albert Einstein won the award in physics for photoelectric effect. Who knew that 100 years just went by in a blink of an eye? Similar to the previous slide, here are the top 15 significant genes between flow groups for smooth muscle cells. Again, further data mining can be conducted to better understand relation between flow and gene expression. For now, we'll focus on the genes related to cardiovascular remodeling. We selected a few genes to examine closely between the groups. For endothelial cells, we looked at CDH5, MMP10, and E-selectin. The OSI groups showed increase in expression of CDH5, which may suggest an increase in maintaining lateral endothelial junction due to loss of cell contact from exposure to flow. For MMP10, we see an increase in expression in the OSI groups as well, and this may be an indication of vascular remodeling promoted by inflammatory stimuli. In the E-selectin marker, it is generally a sign of endothelial activation, in which may be associated with endothelial dysfunction. Here we see a gradual increase in E-selectin genes as the OSI increases. For smooth muscle cells, we looked at ACTA2, myosin heavy chain 11, and taglin. In normal healthy arteries, smooth muscle cells are spindle-like in shape. They are also densely packed with microfilaments and express these three proteins, which are required for their contractility. Here we see these three genes are downregulated in the 0.25 and 0.5 OSI groups, and that could indicate a loss of smooth muscle cell contractility. To summarize AIM-1, we can see that RNA sequencing provides potential protein expressions and changes in cell phenotypes. Dysregulated RNA transcription can lead to diseases. OSI groups exhibited changes in pro-remodeling expressions in endothelial cells and loss of contractility expressions in smooth muscle cells. In the next AIM, we continue our observations on effects of OSI on valvular cell types. We took specific cues from AIM-1, our unbiased RNA analysis approach, and in AIM-2, we are focusing on endpoint responses and genes of interest. We evaluated paracrine signaling events from VECs exposed to low, moderate, and high OSI conditions that lead to VIC calcification. Previous studies conducted by Assad, we looked at OSI models of a diseased valve. After removing the calcific plaques, we found that the original healthy valve regions exposed to high OSIs were where the calcific deposits formed. Another study conducted by Danny involved images of calcified valves from patients with CAVID. The calcific regions were mainly on the fibrosa layer, which was mostly exposed to flow oscillations compared to laminar flow on the ventricularis side. To continue with our cell studies, we purchased commercially available rat VECs and expanded them in the recommended media. The VECs were then conditioned in a bioflux shear assay system with Alexi's assistance using 24 well plates similar to AIM-1. A shear magnitude of 1 dyne per centimeter squared was applied to the endothelial cells for all flow groups for 48 hours. The flow groups were static, steady flow, 0.25 OSI, and 0.5 OSI. We then purchased commercially available rat VICs and expanded them in culture. Similar to AIM-1, we used the conditioned media from VEX in all flow groups to further culture VICs in static for 7 days. 50% of the media for VICs were the previously collected conditioned media from VEX in the bioflux plates, and the other 50% was fresh DMEM or fresh pro-calcific media. The pro-calcific media allows us to examine calcification as an endpoint assessment beyond gene expression. The pro-calcific media consisted of DMEM with 5% FBS, 1% pen strip, 1.8 millimolar of calcium chloride, 3.8 millimolar of sodium dihydrogen phosphate, and 0.4 units of inorganic pyrophosphate per milliliter of media. We chose these ingredients for pro-calcific media because previous studies conducted by Amanda used this pro-calcific media to culture native porcine valves, and significant calcification was observed after alizarin red staining. To continue with our cell studies, we then mixed this pro-calcific media with VEC conditioned media from all flow groups to further culture VICs. The results from this study show that a combination of high OSI with pro-calcific environment leads to VIC calcification. 3 samples were conducted for each flow group, and alizarin red stains to the right show highest calcification in the 0.5 OSI group in pro-calcific environment. The fresh PC group were VICs conditioned in pro-calcific media with no communication from VEX. The results show significant differences between 0.5 OSI and all flow groups except for the fresh PC group. This may indicate that presence of VEC tend to decrease VIC calcification, and that VECs exposed to 0.5 OSI could be associated with VEC dysfunction. Presence of paracrine regulation from VECs seemed to generally lower gene expressions associated with osteogenic differentiation of VICs, specifically TNAP and RUNX2. There was also an increase in MMP2 and BMP2 in the 0.5 OSI groups, which are associated with remodeling. An upregulation of type 1 collagen expression was observed in the 0.25 OSI. This OSI is also interestingly close to the sweet spot OSI. Next, we conducted a similar study with human cells. Human VECs were commercially purchased and expanded in the proper growth medium. The VECs were again conditioned in a bioflux shear assay system at 1 dime per centimeter squared for 48 hours under the same 4 flow groups. We then collected the VEC conditioned media from bioflux, which I will now call this the original group. We placed a portion of this media in an ultracentrifuge to obtain a pellet of exosomes, which are then isolated by removing the supernatant. The exosome pellet was resuspended in fresh pro-calcific media, and the pro-calcific ingredients were also added to the original group as well as the non-exosomal supernatant group. The pro-calcific ingredients consisted of the same ingredients as the previous section to induce VIC calcification. So now we have 3 groups, the original, the exosome pellet, and the non-exosomal supernatant groups. Next, we purchased commercially available human VICs and expanded them in flasks. We then used the conditioned media from VECs in all flow groups to further culture VICs for 7 days. The VICs were conditioned in static in the 3 groups of pro-calcific VEC media that were collected and isolated earlier. We then used alizarin red to quantify the level of calcification in each sample, and the samples were normalized to the amount of protein secreted in the individual wells. The fresh PC group were VICs with no exposure to VEC-conditioned media. The negative controls were VICs exposed to VEC media but with no pro-calcific ingredients. In the original group, lowest VIC calcification was found in the 0.25 OSI group, which is also significantly lower than the fresh PC group. In the exosome pellets, there is no significant differences in the VIC calcification. In the non-exosome supernatant group, we see a significantly decrease in calcification in the static, steady, and 0.25 OSI groups, and a significantly increased calcification in the 0.5 OSI group. To summarize AIM-2, the study included two species, rat and human valvular cells. 0.5 OSI resulted in VEC communication that induced highest VIC calcification. The non-exosomal supernatant group significantly increased VIC calcification under exposure to high OSI. In the next AIM, we continue our observations on effects of OSI on engineered valve tissues cultured in a bioreactor. Based on results from AIM-2, we identified that high OSI paired with pro-calcific environments lead to substantial VIC calcification. In AIM-3, we would like to scale up from two-dimensional cellular studies to three-dimensional tissue culture and recreate the etiology of aortic valve calcification in a bioreactor. But first, what exactly is a bioreactor? According to experts in the field, a bioreactor is a device capable of creating the proper environment for the creation of a certain biological product. A bioreactor must then create an environment consisting of mechanical or electrical signals to condition the biological product. If the system is in static, it is not a bioreactor. Our bioreactor goals include to generate a controlled oscillatory flow environment to grow calcific valve tissues, to provide mechanistic and longitudinal studies of CAVID, and to assist with drug discovery that slows down the progression of valve calcification. We designed a bioreactor that houses 3 valves in the chamber. Prior to fabrication, CFD simulations were conducted with Paulina and Assad's assistance. We created a square inlet waveform, which is similar to the waveform in our lab's previous studies conducted by Dr. Brittany Gonzalez with the torpedo bioreactor. With the input square waveform, the CFD results show low shear stress and high OSI on the valve surfaces, and this flow environment was used to induce valve calcification in combination with pro-calcific media. The images here are different view angles of our 3-valve bioreactor. We used acrylic for our material, and the design was fabricated in Mr. Ziccarelli's machine shop at FIU. The bioreactor was connected to Vivitro's superpump, which is controlled by the Vivitest software with our input waveform that generates high OSI environments. We used pro-calcific bioreactor media to grow the tissues for 7 days. The cells were seated on a scaffold prior to bioreactor conditioning. We chose a bioscaffold for cell seating. We used pieces or porcine small intestinal submucosa as our scaffold type. Why? Previous studies in our lab, we seeded human bone marrow stem cells in both pieces bioscaffolds and PGA-PLLA synthetic scaffolds. We discovered a higher concentration of elastin in the pieces scaffolds after conditioning. and elastin is known to enhance cardiovascular regeneration. We submitted a publication on the elastin-rich bioscaffold valves. This is the abstract for the submission. Critical valve diseases in infants have... the paper was accepted last week and officially published today. Congratulations to all the authors. Besides the elastin content, PCIS is also an FDA-approved biomaterial for tissue repair. It has an acellular extracellular matrix, and it comes in cylindrical shape with no seams or stents. In order to hold the valves in place, we 3D printed valve holders and sutured the PCIS valves on the annulus and the 3 posts. The VECs and VICs were then seated on the valves after suturing and placed on a shaker with seating media for 8 days. The cell seating media consisted of DMEM with 10% FBS, 1% pen strip, 82 micrograms per milliliter of ascorbic acid, and 2 nanograms per milliliter of basic fibroblast growth factor. The seating density was 2 million cells per 2.5 centimeters squared. Since this was a co-culture system, the seating ratio of VICs to VECs was 34 to 27 based on the publication to the right. The VICs were seated first for 4 days, followed by VECs for 3 days. Bioreactor conditioning lasted for 7 days, and the conditioning media used was Bioreactor Pro-Calcific media. It is a combination of the regular bioreactor media, which was used in our previous bioreactor studies and for cell seeding, plus the pro-calcific ingredients that was used in AIM-2. The bioreactor valves were removed after 7 days of conditioning. We also conditioned 3 valves in static using the same bioreactor pro-calcific media for 7 days. After valve conditioning in static and bioreactor under pro-calcific environments, we fixed tissues in 10% formalin and conducted hydrodynamic assessments. We used the Vivitro pulse duplicator to assess hydrodynamic performances of the conditioned valves. The Vivitro system consists of an atrium, a ventricle, and an aortic load assembly, and our valves were tested in the aortic location. The system uses 0.9% saline solution, and the volumetric flow and pressures were obtained using a flow probe and 3 pressure transducers in the aortic, ventricular, and atrial chambers. The Vivitro system can be used to test both left and right sides of the heart. We used parameters for the left side heart conditions: a heart rate of 70 beats per minute, stroke volume of 71 milliliters, a mean arterial pressure of 100 millimeter mercury, and an input 35% systolic, 65% diastolic waveform. Assessment criteria included regurgitation percentage, transvalvular pressure when the valve is in the open state, volumetric flow rate, and the effective orifice area. We tested our static PCIS valve, bioreactor PCIS valve, raw PCIS valve with no conditioning, and clinically available mechanical and bioprosthetic valves. The graphs show each of the parameters from the Vivitro system for all 5 valve types at n equals 3 per valve type. We can see that all valves ranged closely despite the significant differences. This indicates that the raw PCIS, the static PCIS, and the bioreactor PCIS performed similarly to the clinically available mechanical and bioprosthetic valves. We now further examine the 3 PCIS valves. the raw pieces valve, the static pieces valve, and the bioreactor pieces valve. Ideally, we would like the regurgitation factors and transvalvular pressures to remain low, and the effective orifice areas and the volumetric flow rates to be high. These are the clinical definitions of valve stenosis. Regurgitation rates less than 15% are considered mild. Transvalvular pressure of over 30 mm mercury are considered stenotic. The EOA is indexed to the body surface area. In terms of regurgitation, the valves are mainly below 15%, except for maybe the raw pieces valve, which is slightly higher. In terms of transvalvular pressure, all values are below 30 mm mercury. Based on the hydrodynamic results, there is no significant differences in the volumetric flow rates, despite other parameters being significantly different. Lowest EOA was found in the static group, which corresponds to the highest transvalvular pressure and highest jet velocity. Highest regurgitation factor was observed in the raw pieces valve. Overall hydrodynamic results show that all valves are not stenotic. We then conducted nanoindentation to further examine the mechanical properties of the valves. Mechanical assessments using nanoindentation was conducted by Dr. Lowe from Dr. Agarwal's lab in the MME department. Highest Young's modulus was found in the static group. The raw pieces, or the unconditioned group, exhibited the lowest Young's modulus. All three groups were significantly different from each other. From the nanoindentation and hydrodynamic data, we see an increase in valve stiffness, which leads to loss of flexibility and poor coaptation. Loss of leaflet flexibility results in small EOA, and small EOA leads to high jet velocity and therefore high transvalvular pressure based on the modified Bernoulli's principle. Here we see a trend on the three graphs correlating stiffness and transvalvular pressures. The higher the stiffness, the higher the transvalvular pressure. The valves were then cut and embedded in OCT for sectioning. We embedded the valves in transverse and frontal planes and sectioned them at 16 microns with a cryostat. The section pieces were stained for calcification using Alizarin red. Alizarin red staining was conducted with Andres' assistance. We can see here that the bioreactor and static valves had calcified regions dispersed throughout the tissue, and no calcification was observed on the raw pieces valve. The images were then quantified using Assad's MATLAB heat map scripts. The level of calcification was higher on the bioreactor valves compared to the static groups, however, not statistically significant. We then conducted immunofluorescence stains to further examine the distribution of cells and specific proteins. We stained for alpha SMA in red and CD31 in green in our immunofluorescent assessments to further examine the distribution of proteins in our tissue samples. We quantified Z-stack images of static and bioreactor samples, and we found that the bioreactor group had a relatively higher expression of both alpha SMA and CD31, though not statistically significant. In summary, our AIM 3 showed that bioreactor can be an effective device for valve tissue engineering. Our hydrodynamic assessments show a correlation between high Young's modulus and low EOA and high transvalvular pressure. Alizarin red staining show early onset of calcification with bioreactors slightly more calcified than static valves. Despite the fact that bioreactor conditioned valves had higher calcification, the hydrodynamic assessments show better performance in bioreactor valves compared to statically calcified valves with a significantly higher EOA and significantly lower transvalvular pressure. Calcification levels of static Pisces valves and bioreactor Pisces valves are similar to AIM-2's VIC calcification from the original media group. Immunofluorescent stains of both alpha SMA and CD31 in static and bioreactor valves were not significant. VECs and VICs were evenly infiltrated and dispersed despite VICs being seated first for the first 4 days. Special thanks to Assad for assisting with image quantification. Some of the study limitations include choice of pro-calcific media may alter results of mechanistic studies. Certain ingredients may alter gene expression results. There is no significant correlation between calcification and immunofluorescent stains. The valve holder in the bioreactor is not completely secured in place. Future studies include further data mining on RNA sequencing results, immunofluorescent staining for other early cavid proteins such as GAGs, conducting protein analysis on conditioned media using mass spectrometry, assessing the valve opening and closing geometric orifice areas from high-speed camera videos, and analyzing valve closing time between healthy and mildly calcified valves. The videos shown here are high-speed images of 2 different Pisces valves. Can you tell which one is raw Pisces valve and which one is the bioreactor conditioned Pisces valve? I'd like to thank my major professors, Dr. Ramaswamy and Dr. Hutchison, my committee members, Dr. Sukias, Dr. George, and Dr. Aikawa. Thank you, collaborators from industry and other departments, colleagues from CMRL and CV Putix Labs, teaching assistantship from the BME department, DYF from UGS, and all of my friends and family members. Special thanks to Claudia Estrada for helping me reserve a room for my dissertation, as well as all past conference registrations, purchase orders, and many, many more. Also, if you enjoyed the cupcakes, please follow Joyful Cookery on Instagram. Thank you, everyone, for your attention.